Okay, welcome everybody. Welcome to the New York Genome Center and welcome to the VIZBI showcase. My name is Christian Stolte. I work here at the New York Genome Center as a data visualization designer. And that job description or that job title has sort of the three key words for tonight's event in it. Data, as in the evidence that we collect at the beginning of any scientific project, or what we generate in the course of a scientific project. Then visualization, which is either making that evidence visible or showing the results of the research in some way that allows people to relate to it. And then finally design, which is what makes the bridge to understanding. How do you communicate all those things that you found in your research effectively and efficiently? Design can help a lot in that. And so tonight, we're going to show you some great examples that came out of a conference series called VIZBI, Visualizing Biological Information, that Sean O'Donoghue is going to talk about later. But we're switching the program around a little bit, and so I want to go straight to our keynote speaker for the evening, Gaël McGill. He comes to us from Cambridge, Massachusetts. Originally, he's from France, but we won't hold that against him. And he works at Harvard Medical School and also has his own company, Digizyme, where they produce animations for the life sciences. And he's, I would say, one of the pioneers of molecular animation and a microbiologist by training?
Molecular biologist.
Okay.
Mm-hmm. Great.
Molecular biologist.
Same difference.
That's right. Anyway, so without further ado, I'll have you, Gaël McGill.
Thank you. Thank you, Christian. Can you all hear me okay? Great. Well, it's a privilege to get to share one's work, so thank you for coming and spending the evening with us. Let me switch over to the presentation. So as Christian mentioned, I kind of split my life as an academic on the one hand, as an investigator, as a faculty at Harvard Medical School, but then for a lot of the projects that I'll show you tonight, that's if you don't fall asleep on me in the cozy darkness of this room, are actually produced as part of my company Digizyme. And we work with a number of different clients, from science museums to medical research institutes to publishers, pharma, biotech. So let me start with an anecdote, I guess. So about 6 months ago, I had the most terrifying teaching experience in my life. I'm used to graduate students normally, but in this case, I was in front of about 25 sixth graders. Yes. So for anyone who's experienced that kind of an audience, you know what I'm talking about. You have about 20 seconds to engage them, or judgment will be passed and you're in trouble. So I decided to start with an animation of a bug. I figured that would capture their attention. But this is no ordinary bug, and it's also not an animation, actually. It's an interactive, and it's part of a project that I'll mention a little bit later. But, so it allows them to explore some of the anatomy, right? They can hit some of the different buttons. I then had to introduce a couple of big words. Remember, these are about 10-year-olds, right? Hydrophilic, hydrophobic. The purpose was to get them to consider that this is not any ordinary beetle. This is the Namibian beetle that lives in one of the most arid places on Earth. And it likes to climb up the dunes in the morning and stick its behind up in the air. At this point, I had their attention. And the fog washes over this creature, and because of its hydrophilic and hydrophobic patches on its back, it is literally able to collect water from the sky. There's no standing water. And it turns out that this type of technology, this biomimicry, is what allows us to create technologies that allow to bring water to human populations in deserts as well. So what I was trying to do with these eager young minds was to kind of shift their mental model of what their, typical beetle that they might see and crush in their backyard, and reconsider that in certain species, maybe in every species, there are unique engineering secrets that nature has already time-tested for us over millions of years. And they're not only things we can learn, but they many times turn out to be inventions that lead to more sustainable engineering practices. So I just mentioned mental models, and let me take a minute to define that, because really what I'll show you tonight is really centered around the idea of how do we use visuals to understand science, but also how do they shape our mental models of science. So what do I mean by mental model? Octopus. I'll let that sit with you for just a second. So you're all processing that in your own minds, in your own way, right? And I would venture to say that there are as many different mental models of an octopus as there are people in this room, right? So if you're thinking of something like this, I don't know, maybe if you're a marine zoologist, you see this. Maybe if you're one of those middle school students, you see this, or more likely this. But what if you're a nature photographer, or a neuroanatomist, or maybe a computational biologist, right? So everyone's going to think of this in a slightly different way based on their own experience, and since we're close to Wall Street, right? So octopus means a lot of different things to different people. And I'm only, part joking, because if you really consider, for an octopus, it's one thing. We may have all had an opportunity to stand around an octopus and observe the macroscopic object together. And if we did that, we might align our mental models a little bit better. But there's still a great variety of ways that people think about these depending on their experience and kind of their cognitive filters. So what happens when you try to break down the process, when you try to better understand some of the steps that go into forming mental models, right? So there's not only the visual input, but there are these cognitive filters, and there's, of course, pre-existing knowledge, past experience. And each one of these steps has the potential to be different between people. And I won't go into the details of that, but even for a new concept, there's no such thing as a tabula rasa. You're always trying to connect information to pre-existing experiences. So that's octopus. But I would venture to say that when we start to say things like insulin, now we're really in trouble, right? Because insulin is not something we can all gather around in an aquarium, point to and look at and observe together. We can build models of it, but that's exactly my point. And it gets worse if we say ribosome, or transcriptional enhancer, or mitochondrion, or cell. In fact, cell is a big problem. So back to the 6th graders. This was a day I had in the Brookline Public School, so I had these 5 groups of 6th graders all to myself, and they were studying cells. So I figured I would meet them in the current lesson that they were learning about, and so I put up a very simple diagram of a cell with the words that they had been asked to learn at their level. But then I wanted to scare them a little bit. So then I did this, and I said, in a few years, you're going to have a few more words thrown at you. And then it's going to look like this. At this point, they were kind of freaking out. And then it's going to look like this. And then like this. And then I told them that this was basically the kindergarten version of the cell. And again, I had their attention at this point. And my mini experiment to let you in on what I was trying to do is, as I'll show you in some of our studies, we're very interested in understanding how to design visuals that give our audiences an intuition for the real complexity of living systems. So after telling them this was the kindergarten version of the cell, I threw this at them. And this is an image that we created a number of years back now, but every protein, every nucleic acid, every structure that you see in this image is actually based on real X-ray structures in the protein data bank. So the actual geography of the elements is a little bit curated, right? So one could take a slice from a technique called cryo-EM tomography, which literally gives you the map of a piece of a cell. But this was more of a didactic image, and I'll come back to it a little bit later in the talk to show you how we turned it into an interactive. For the students, I just wanted to throw this complexity at them and see what happened. Did I put them to sleep? Or luckily what happened was the other thing, which is that they all raised their hand, which is exactly what I wanted. They had questions. They were a little bit hooked. The next thing I threw at them was this, the fact that in just a tiny little piece of this cytoplasm, this is actually what things look like. This is a piece of a relatively famous coarse-grained simulation now that Adrian Elcock did on E. coli cytoplasm, where he took a recipe of the 50 most abundant proteins in E. coli and did a steric, hydrophobic, and electrostatic coarse-grained simulation. So I wanted to give the students a sense for what some of this complexity, not only the geography and the crowdedness, but also the dynamic motion. So this was also a bit of an introduction into basically our work into education. So a few years back, we were fortunate enough to be able to partner with Apple. This was just a few months, unfortunately, before Steve Jobs passed away, but just in time to kind of get the project signed off on and approved. At that point, Apple was interested in really seeing what the iPad could be used for in education. And we were fortunate enough to be the team that they selected to work with to create a new textbook from the ground up. So not an adapted textbook that already existed in print, but to really start things from scratch. And so I'll show you a few examples of the project. The project itself was meant to target high school biology students, and it was molecules to ecosystems, so the full high school curriculum. And we wound up creating hundreds of images, animations, interactives, even in certain cases, small games for this textbook. So in certain cases, and related to the experience that I gave the sixth graders that I showed you already, we wanted to bridge the gap between the typical textbook figures that have these kind of empty cellular compartments and give students the ability to, in this case, concentrate that cytoplasm and look at different elements within it. So this was not a movie, this was an interactive where using your finger you could change the density of elements in the cell. We would also leverage some of these landscapes. There were buttons on the left to help map some of the molecular families to different regions of the cytoplasm and give students a sense for the relative abundance of the elements. And remember, this is high school, so the goal, the learning objective is not for them to remember each and every single part of these processes or different cellular geographies, but to get a feeling for molecular families and cellular morphology. We also developed digital paintings like this one, where with very simple interactives, by tapping the buttons below, they could observe different phases, developmental phases, in this case, in spermatogenesis. So digital paintings, 3D, in this case, anatomical sculpts. So these are 3D models. Some of them interactive, where we wanted students to understand how certain types of joints, and we related them to kind of engineering analogies. So the students could either tap the buttons in the upper left, or they could tap the different areas in the skeleton. And again, each one of these, as I'll mention a little bit later, is carefully crafted with very specific audience and learning objectives in mind. I think hopefully what you'll get from my presentation tonight is that the minute you make any image of science, you're by definition making an interpretation, a model. And my favorite quote from my grad students is always, all models are wrong, some are useful. I think that's a great way to think about things. So keep in mind as you see these images that at least in the way they were designed, they're always with a very specific audience in mind and very specific learning objectives. This was another interactive that helps students navigate different concepts in viral structure. And then this was actually one of our more popular interactives. And we went to high schools before starting the textbook and during the testing of it to ask teachers of high school biology, what are the toughest concepts that you run into with your students? One of them was reproduction. And one of the reasons in particular for the menstrual cycle is that the teachers said they were having trouble relating what is really 6 or 7 dimensions of information and explaining to students how all of these change simultaneously. So instead of making a linear narrative, a movie, to explain to the students, we thought this would be an opportunity again for an interactive. So as you move your finger on the iPad and pick the date of the month in the bottom, you're able to observe some of the different changes, some of the hormonal levels and how they relate to either the release of the egg and the anatomy of the uterus. And I'll come back to this theme of when is it appropriate, depending on audience and learning objective, to do a static image or even no image at all. When is a paragraph of text better suited to a particular communication objective versus an animation or an interactive?
For infectious bacteria, host tissue is like a petri dish of rich culture. Bacterial populations can grow exponentially on host nutrients, doubling as rapidly as every 15 minutes. Antibiotics interrupt the growth cycle and kill bacteria while the host's immune system catches up and eliminates the infection. One of the most urgent problems in medicine is the emergence of antibiotic resistance, which is actually promoted by antibiotics. A bacterium acquires resistance to an antibiotic through mutation of a gene or transfer of an antibiotic resistance gene from another bacterial species. The prevalence of a rare gene with neutral adaptive value would remain very low in a normal bacterial ecosystem. However, antibiotics impose massive selective pressure on bacterial populations, killing off the bacteria that do not have the resistant genes. Resistant bacteria can grow exponentially when all of their competition is wiped out, and antibiotic resistance can then become widespread.
So, of course, we also did use linear narratives, animated stories, in ways that we hoped also made the science memorable to the students. They all tend to, freak out when the little green balls spread out over the screen. They remember it. So we use animation, but also simulation in different ways. I believe the next example is one where we wanted the students to understand that even though they learn about the typical bilayer membrane of cells, there are some very unusual archaeal cells that live in extreme conditions, like deep sea vents, at temperatures that would normally burst apart cells that have membrane bilayers. So if you look at the structure of some of these, the lipids here on the right, you notice that it is still technically a bilayer, but they've actually fused the hydrophobic tails of what are normally our lipids. So it's a monolayer, but structurally it looks like a bilayer. So we combine static imagery like this with the following simulation.
The archaeal single-layer membrane is an adaptation to high-temperature environments where only thermophiles are found, such as hot springs, volcanically active fissures, and deep sea vents where superheated water gushes upward. The single-layer membrane maintains its structure at near boiling temperatures that would disrupt the structure and function of lipid bilayer membranes.
So this is an example of an actual molecular simulation, of a molecular dynamic simulation. For those of you who may be familiar with the technique, we're still able to do these types of simulations on our cloud, and it's typically not the kind of simulation that is used in an educational context, especially not in high school. But our approach is that we think it actually is a great way to illustrate what might be the difference between these two structures. So the Life on Earth textbook, which by the way is out there and freely available, so we found that it's gotten use way outside of the high school target audience. So if you're interested, I encourage you to go download it. It's available in 7 different books from the iBook store. And we've continued to experiment with ways of using interactive media to teach different aspects of science. So this is a book, interactive book on coral reefs, which is still in production. In this case, we've used these landscapes are about 3 or 4 iPad screens worth of width that you can navigate and launch mini documentaries along the way. So we're trying to play with the notion of what a linear book feels like. Another project is Nature's Toolkit. Back to the Namibian beetle that I showed you, that was actually an interactive from this book, which is based on not only biomimicry, but the idea of sustainable engineering practices. And this is a project that is a science museum exhibit, as well as a digital book and a STEM curriculum. So let me take a few minutes to give you a sense of our work more broadly. I'd like to just take a few minutes to play a sort of education reel that gives you samples of some of the work that we've done. Sound okay?
Each probe group consists of millions of identical single-stranded probes. A single-stranded probe matches part of the sequence of a gene in an organism. To measure gene expression using a microarray, the mRNA in the cell is isolated and amplified. Complementary DNA, or cDNA, is synthesized from the single-stranded mRNA using an enzyme called reverse transcriptase, which copies RNA into DNA. Sequener enters an echinoderm through a pore in the skin called the pancreate, travels to the root canal, and makes its way down the radial canals of each arm, where it enters hundreds of tube feet. Movement of the tube feet is controlled by opposing muscle pairs. Muscles in the ampulla, the bulbous top of the tube foot, contract, while opposing muscles in the base of the tube foot relax, and the foot lengthens. Signals from about 30 different photoreceptors are linked by natural selection with animal sources of protein for egg production. Feeding consists of several behaviors. A head knob pierces the victim with a channeled blade. Saliva is injected that blocks blood clotting. A tubular mouth organ draws out blood. The feast concludes with a signal that the stomach is full, and an initiation is late. The abortion mosquito rests its tail down on Gargle's chest and squeezes the water from the blood vein. This behavior is an important part of mosquito control. Careful treatment with calls and active mosquito nets are among the most reliable ways to eliminate mosquitoes, which are the effective collective organisms long enough to absorb lethal doses of insecticide. The plasma membrane of animal cells, for example, is rich in cholesterol and a fresh variety of receptors on channels that capture and transduce extracellular signals. The rough endoplasmic reticulum membrane supports the synthesis of non-cytosolic proteins. Here, black cells stop endopeptide channels that generate weakly translated polypeptide chains through primary reticulum. In contrast, the inner membrane of mitochondria is packed with molecular machines cytoplasm with radiocarbon ingredients. Molecular turbines, ATP synthase, harness those ingredients to synthesize ATP. In each of these examples, the composition of the lipid metabolites of various lipids and proteins is different and supports the unique functional compartments of that membrane.
The following is from our Coral Reef iBook. Some of the examples of those mini documentaries that I mentioned.
Parrotfish are essential to the health of coral reefs because they feed on algae that goes into the coral. They are specifically named for their parrot-like beaks that are strong enough to bite through the line-strung skeletons of fruit. Parrotfish are generally considered herbivores,
Okay, so hopefully that gives you a sample of some of the different technologies and kind of design approaches that we use to make some of the material more engaging for various audiences. I mentioned earlier that we also do a lot of work with the life sciences industry, so I thought I would show you just a few examples of some of that work. So in this case, you know, some of the tasks, some of the more complex tasks of molecular modeling, even for science groups within pharmaceuticals, in this case Genentech, we created an animation that explains some of the molecular mechanisms that kick off the cell suicide process, apoptosis. In this case, the FAS-induced apoptosis process. So either animations or sometimes really abstractions. This is a short sequence we did for a CEO who was having trouble explaining Bayesian networks with kind of math. So he wanted a visual proxy for some of the things that their algorithms are doing. So obviously the accuracy for a piece of this is taken down and moved away from the mathematics, but we hope to give the audience a sense for some of the different filters and selective processes that are used in their network building algorithm. All the balls fall into a magic box from which fall out the networks. I leave it to you to decide if it's oversimplified, but that was the task. I'm going to skip over this one. We do, of course, also a lot of work for marketing and communications of these organizations or these companies. In some cases, since a lot of the work we do borrows from the tools of Hollywood and the entertainment industry, but is adapted for science visualization, and I'll show you some of the software we've developed. Sometimes those skills are called upon to model and design virtual instruments. So in this case, we worked with a couple of different companies to create instruments that were 2 to 3 years away from actually existing in real life. But the images were used not only for their marketing, but also to rally the engineering team around a prototype, a visual prototype. And then sometimes we're involved in actual product launches. And again, in the interest of time, I'll just skip forward, but you get the idea. It's these photorealistic instruments that are being used as part of a product launch to explain how the instrument works. So back to the molecular world, because that's really my area of training as a molecular cell biologist, and also where I think some of the unique challenges of depicting the life sciences exist. Certainly there are some all the way up and down the temporal and spatial scales, but my particular interest is the molecular world. So what is so difficult about the molecular world, and why do students have a hard time with it? Why do all of us have a hard time with it? Part of it, as I mentioned already, is the crowdedness. Some of it is also the notion that, again, to create any image of anything, it is already an interpretation, whereby one can take a data set and then select through a number of different visual representations, depending on what you're trying to say, based on that data set, different molecular representations. It makes me think of, probably people are familiar with this. Well, it's the same thing for proteins. I had to slip in some French in there, right, to fight back. So there you go. Ceci n'est pas une protéine. So physical scales, right, that's an obvious one. We have zero intuition for how things behave at the molecular scale. Things like gravity, doesn't matter. I mean, it's there, it doesn't matter. Things like color, right? One of the favorite questions I get from my grad students is, what color should we make the DNA? It's like, there is no color. It's below the wavelength of light. Color has no meaning down there. So I'm not saying don't use color, but use it in ways where the design objective are serving a particular purpose. This is another example of trying to bridge scales between the macroscopic and the molecular, where high school students, I'll just let it play for a second, sorry. I'm guessing there are a lot of these run here in this building, probably. But the idea there was to give students a sense. This was for high school students who were experiencing their first molecular biology lab experience, and were given, a few clear tubes to pipette with, and have no context for what they're doing when they're loading, for example, an agarose gel. So some of these animations are meant to help them bridge that gap between the macroscopic and the molecular. This is kind of a funny sequence we were asked to do by Life Technologies to visualize how the evolution of their sequencers might look like over a period of years, and how one might eventually go to a doctor's appointment and have a sequencing machine right there on the doctor's bench and have quantum dot-based sequencing. So again, the theme here being bridging the gap between the macroscopic and the molecular. We've also used some of our tools, which I'll come to in just a few seconds, to help specific fields. In this case, in synthetic biology, a subfield of DNA origami. These are scientists who are using DNA not as just an information storage molecule, but as a structural building device. So creating more and more complex shapes that self-assemble with DNA. This is an example actually of a device that was designed by one of my graduate students, Sean Douglas, who's now at UCSF. It's basically a hat shell design. So you have two sides, which will be colored in just a second. So this is one side of the clamshell, and then the other side colored in red now. In the back are two small hinges, again, made of DNA. And in the front is a latch system of double helices. And then inside is basically a payload, in this case, a therapeutic antibody payload. So the idea is that in the presence of a particular molecule, which in this case is PDGF, those latches in the front are actually aptamers, meaning that the nucleic acids and the double strand will be competed away in binding PDGF. And only in the presence of the PDGF is the latch unfurled and the clamshell opens up and reveals the therapeutic payload. So these types of nanorobots, if you want to call them that, are based on DNA self-assembly, DNA origami. And with existing science visualization tools, it's actually quite a challenge to create these types of models. So this is where some of our software came in handy. And then temporal scales as well. So explaining to students at the molecular scale the fact that not only are there dynamics, but there are dynamics that are happening on multiple scales of space and time simultaneously. And then, so a particular detour with stochastic random motion, because this is actually one of the things that we're studying as part of our, as part of my academic research. So, back to the sixth graders, or again, frankly, to anyone, to understand that within each, let's say, eukaryotic cell, there are 10 billion proteins, and that within the space of every second, every molecule encounters every other molecule at least once. I love saying that because I usually see people's minds kind of go, and it does the same thing to my mind every time I say it too. It's just, again, it's outside the scope of human intuition. So how do you get across to students, how do you give them an intuition for the fact that these interactions are kinetically random? And this is where some of our kind of cognitive psychology studies are coming into play. And a lot of the work that I'll present here is in collaboration with Jody Jenkinson at the University of Toronto. So we've tried to build adaptive decision trees to try to understand exactly what are the misconceptions that students have, in this case, undergraduates. And this is yet to be published, but what we've already found with a sample of over 1,000 undergraduate students is that over 80% of them think that molecules know where they're going. In other words, molecules literally self-propel to go meet wherever their binding partner is. I don't know about you, but to me, that's a big problem, right? On the one hand, we observe kind of the efficiency of biological systems. But at the molecular scale, we have trouble reconciling that with the fact that molecular motion is random. So I won't take you through the entire decision tree, but you can see some of the questions that we're throwing at them, and you can see the breakdown. This is actually a visualization of the results from the students. That this is the 83% right here. And so depending on what they answered in the first question, we try to refine and gain a better understanding of what are they actually thinking. I'll give you another teaser before the paper comes out, which is that a lot of the students in follow-up focus groups, we found out that if you ask them what happens to molecules while the cell is kind of inactive, again, whatever that means, molecules, no, then they're random, they're hanging around. But when the cell receives a signal, so when there's some sort of a signaling cascade that's triggered, then the proteins know where they're going, right? As if there was some sort of a tractor beam that gets kicked in. So there are some pretty serious misconceptions, and we're trying to understand what the problem is. Part of the problem, we think, is this. So what kind of mental model of molecular motion do you think students are getting if you see an animation like this? And by like this, what I mean is that the hormone, in this case stem cell factor, is gracefully landing on the two receptors that come and welcome it and know exactly when it's about to land. This is actually, I would venture to say, the majority of medical animation falls into this category, using the 12 principles of animation from Disney. Overlapping motion, ease in, ease out, all the stuff that you learn as an animator. So we wanted to try and see what happens to students in certain specific tests if you don't show them one version, but if you take, let's say, 120 students and you break them up into 4 groups and you show them 4 different versions of the same animation, but in certain cases with a lot more supporting actors, in this case actually molecular water. This is basically the version that educators would probably tell you you're out of your mind. Why would you ever show this to a student, right? Well, if you also do eye tracking on the students while they're seeing these animations, you actually learn some interesting things. The first thing you notice in certain cases is that they get bored. So watch what's about to happen here. This is my favorite part. They actually start to watch the QuickTime cursor to see when this thing is going to be over. But the more interesting thing is if we can look at the eye tracking in the more complex versions, again, depending on how these things are designed, if you still allow them to follow the primary action, they do just fine. In fact, not only they do just fine, but they score better on tests that are asking them questions that go beyond the purely factual, does A bind B? So we're more interested in questions along the lines of, what happens when you increase the temperature of this environment? Right? Can students gain an intuition for molecular environments if you show them animations like this? Or do you need to consider bringing in some of the complexity? And the jury's out. I don't have a black and white answer for you, but these are the types of studies that we're doing. And we're trying to come up with kind of, in parallel to the Disney animation principles, why not have molecular animation principles? Things that at least remind animators some of the tools and techniques that might facilitate or rather avoid gaining student misconceptions. And we don't just do it with molecules. We're also doing other processes like how do you teach insect locomotion? What are the design parameters that facilitate students understanding that basically it's an alternating tripod mechanism? Right? Do you want a photorealistic ant rendered in 3D, or do you want some of these simpler models? These are the types of questions that we study. Visualization is powerful, but it's also powerful in the other direction when things are wrong, right? So what do all these DNAs have in common? I already gave you the answer, and this is a genome center, so I'm assuming everyone's already picked it out. They're all wrong, right? In different ways. They either are the wrong handedness, or they have no major minor groove. Why do we care? Well, because these things actually really matter once you start to understand how the machinery of the cell reads DNA sequences and how it regulates genes. Another one of my favorite examples. This is a site which sells accurate 3D models of anatomy. No? Anybody? It's a backwards brain. I don't know. I mean, if that doesn't bother you, I don't know. But, my point, I guess, is that there's no reason for this, right, in 2018. I mean, we have so many tools, so much data, there's absolutely no reason for this sort of thing. And so I'm actually going to skip this for the interest of time, but I'll just quickly mention that what we've tried to look into is whether we can do a better job as a community, visualization community, in providing more data along with our visualizations to explain some of those design choices. So not only where did I get the data, what was based on data, what was based on interpretation, what was just a guess, because there are plenty of those in some of our visualizations as well, but also what were some of the design decisions. And so what we've tried to do is propose a methodology by which we could create annotated visualizations that we think over time hopefully would increase or rather raise the bar of scientific accuracy. So we looked a little bit into some of the things that might lead to a mental model, but I want to end with some thoughts about what happens going in the other direction. In other words, not just how visuals help us understand and communicate science, but one thing I've noticed over the past 10, 15 years in working with scientists, it turns out that if you get scientists to make visuals of their own science, even when they're the world expert on a particular topic, they start to see their data in a new light. It kind of recontextualizes their results. And so what we've really come to realize is that it's not just the endpoint of the visualization process, but it's the process of making those visualizations in and of itself. My own little rule of thumb or experience has been that, and I think it's probably the same for most of you, when you're kind of at home comfortably in your own head thinking about something, that's one level. But the minute you have to, let's say, write it down, the minute you have to write a review, the minute you have to write anything, it's not just a process of taking that information and plopping it down onto a written media. It's an active process. It's a thinking process. It's painful, usually. The next step is, even for those of you who teach, know that you don't really know something until you've taught it a few times. And I think that's the same process. You don't just have to know it, but typically to be a good teacher, I would say you have to be able to explain it 3, 4, 5 different ways for different types of learners. So in this little hierarchy of kind of stringency of thought, for me, visualization comes at the end of that process, which is, it is a very cruel process in that you can't get away with missing data. So if you try to create a visualization of a process or of something, it will instantly tell you where the missing data is. And it turns out that's a really powerful thing to experience as a scientist trying to understand a process. So, all kinds of separate issues come up when you're trying to make visuals. The first is to define who exactly am I externalizing this information for, right? It's going to be very different if I'm making an animation of that beetle for a 6th grader versus for a beetle expert at some beetle conference, right? The other thing is, now we have a number of different choices to make. And I talked about this for the textbook. What is the best vehicle of delivery for a particular piece of content? It's not always the same, right? So you have that choice. And then finally, there's just skill sets involved. Some of these things require software, which is hard to learn. So in the last few minutes, I'll zip you through some of the things that not only we've done, but also some of the tools and resources that I hope to leave you with if you're interested in visualization yourself. So a few examples of different ways of conceptualizing information. This is a cancer poster that I designed about 10 years ago for a company called Cell Signaling Technology. And it was meant to summarize at least what was known back then about some of the key pathways in human cancer. It was recently redesigned with colors that I felt were a little too happy for a cancer poster, but that's my own opinion. I didn't have final say on that design. But other types of, again, kind of infographic posters that mix either kind of a 3D immersive landscape, in this case a tumor and microenvironment, with more detailed structural information. I showed you this image already, but what if you could actually navigate that image? So I'll just take a second to show you. That image actually exists as an interactive online, where you can move your mouse over any one of the proteins and find out what it is. You can also navigate this diagram by pathway. So for example, show me apoptosis, whereby Cytochrome C in purple is being released from the mitochondrion, leads to formation of the apoptosome, caspase 3 activation, and cleavage of the cytoskeleton. So again, it's not just about the images, it's about how you allow people to navigate them and to learn from them. So again, to end, tools and training has been one of the areas of focus. So in terms of tools, about 10 years ago, we started creating a software called Molecular Maya. So Maya is one of the workhorse software from the Hollywood industry. So if you've ever seen, a Star Wars movie or a Pixar, Lord of the Rings, they're all made using Maya. The problem with Maya is that it was never meant for biological data sets. So what we've started doing using Python is to create a series of kits that fit inside Maya and transform its abilities to receive structural data sets and even dynamic data sets. So I'll zip through a few examples. We have a DNA kit whereby students can just draw a curve, click a button, and get structurally accurate and simulation-ready DNA. We have also kind of a RNA single-stranded DNA kit that lets you not only model very quickly sequences of single-stranded nucleic acids, but create dynamic hybridization animations, cloning operations. We have a modeling kit that let you integrate various data sets, in this case, a model of the kinetochore. One of our kits automatically rigs biological molecules. So for those of you not familiar, a rig is basically the virtual armature, like a skeleton, that animators use to make their 3D models move. And we have so much information on how proteins and other molecules move dynamically that we're able to dynamically rig them, again, with a click of a button. These are not tools that currently exist in the scientific community, and we're trying to build them within these 3D applications. A cytoskeletal kit that lets you build large molecular meshes rapidly based on cytoskeletal elements, and then an environment kit where you can provide a recipe of components, in this case blood plasma, and create a coarse-grained simulation of some of these environments, as well as membranes. So Clarify is the resource through which a lot of this training and these tools are available. This is the training page. We have a number of different courses that are offered. Each course is made up of different video chapters. For any of you who are familiar with sites like lynda.com or Pluralsight, this is kind of the same idea for scientific visualization. I'll take 10 seconds to give you an example of a few trailers for our courses. Am I already over time? Or I'm doing okay? So the training really covers the entire 3D pipeline as well as certain data visualization techniques and packages. So that was storyboarding. We have one actually that relates back to our iBooks author process. So if you want to learn more about how we created our textbook with E.O. Wilson and Apple, this is an example of a course that might be of interest to you. And then the final example also is really not just about teaching software and design techniques, but in certain cases we're finding that there are certain parts of the scientific community that inherently have a hard time understanding techniques from other parts of the scientific community. So I referred to molecular dynamics earlier this evening, and we find that a lot of non-biophysics people who are not familiar with computer programming or the math have a hard time understanding what exactly goes into a molecular dynamics simulation. So we've created a course. I won't say molecular dynamics for dummies, but that's kind of the approach, to use visuals to try and explain some of the concepts without going into the math. And I'll play just a few seconds of that. So this course takes us through all the different forces that are being used and calculated during molecular dynamic simulations. Clarify is also meant to be a resource for people who are interested in entering the field. I think probably Sean and Christian would agree. We actually get a fair number of people contacting us who are interested in doing this type of work and who are either scientists or artists by training, but who have a hard time, visualizing, no pun intended, how to get into this type of work. So I put together a series of interviews with a number of different people working in the field, each of whom have kind of their own unique career path into the work that they do. And also there's a collection of kind of curated articles on science visualization. And what we're hoping is that this part of the website is kind of a useful career resource for people who want to do this type of work. So in closing, I think what I've shown you is that in trying to consider not only what are the stimuli and the filters that go into forming our mental models, but also what are the hurdles that we need to go through in creating visualizations. And this is really kind of my own mental model of how I do this work. Some of these questions are kind of tough to ask in a company setting, which is why I do them as an academic at Harvard Medical School. And when it comes time to implement specific projects for clients, it's a lot easier to do it in the context of a company. But the whole process is really what ultimately interests me. And all of our work, as I hope I've shown you, really sits at the intersection of these different areas. One thing I didn't mention, but Digizyme, I hope, is unique in the way that every single member on the team who touches pixels or programs code is also a scientist by training, a PhD level scientist. So instead of just collaborating scientists and artists, and don't get me wrong, there are many wonderful examples of collaborations between scientists and artists. At Digizyme, what we've really tried to do is to integrate the knowledge of the science and of the design and of the production within single individuals. And we feel it gives us a way of approaching the material which is different. So I'll end with two quotes. I promise these are the last two slides. The first one by Max Planck and an illustration by Stuart Jansen from our group, which is really this idea that visualization might help us to bring all these data sets together and to create these mental models of what are otherwise highly complex data sets that come from multiple sources. And then to give the last word to the host of last week's event, in a sense, at the Broad, if a picture is only worth 1,000 words, we're screwed. This is Eric Lander, who I believe in VIZBI 2011, I think, opened the meeting with this wonderful quote. So thank you for your attention, and I'll be happy to take any questions.
Thank you, Gaël.
Welcome.
And now it's your turn. You can ask Gaël anything you want.
We shouldn't have turned the lights down. They all fell asleep on me. That's what happened.
Well, Maybe I can start things off with a question. How
Sure.
did you get into animation, Gail?
Uh,
From starting out as a molecular biologist.
Right. So my dream was always to do molecular biology. So, but I had a lot of art growing up, a lot of art and music. So I majored in those in college. And as a second-year graduate student, even though the science was going great, I found myself really excited when I had to give lab meeting, which is pretty odd, right? Because most people hate to give lab meeting. So that's when I started Digizyme, actually, as a second-year graduate student, where we found that, at least in the Boston area, there were a lot of companies that were in need of scientifically informed design. And we found that a lot of the competing groups were marketing their services to the life sciences, but didn't necessarily have scientists doing the work. So that was of interest to us. So we started very simple, doing a lot of corporate identity. I mean, not that basic design is simple, but corporate logos, branding campaigns. Got into 2D animation a little bit, Flash, right, back in those days, and then we moved into 3D visualization, and really liked the idea that we could create more immersive animations by using the tools of Hollywood. So it was always kind of there, but the epiphany came in grad school. The 14-hour days in the lab were not long enough, so we made pretty pictures at night, kind of thing. So, yes? Yep.
Can you comment about the post-production process, and also did you see this application, maybe not in final detail for this, but maybe research presentation posters, things like that with
Right.
digital signage?
Yeah. So by post-production, you mean kind of the final steps that lead to the visuals that I showed, or?
Like animation with rotoscoping, or are you
Right.
getting your own process as you get this knowledge?
Yep. So, I mean, we didn't just borrow the tools of Hollywood. We've really borrowed their way of doing things as well, because they've figured it out. I mean, they've thrown billions as an industry. A quick note that is, I think, maybe part of the question, or I'll make it part of the question, but I like to say it because I think people often forget that in the Hollywood industry, there's so much specialization. So if you are a modeler, character modeler, or a lighting technical director, and that's all you do all day, as a science animator, you need to know the whole pipeline all by yourself, from pre-production, storyboarding, to modeling, surfacing, rigging, animation, dynamics, rendering, compositing, sound, video editing, the whole thing. And it's both what makes it, I think, really challenging and also really exciting. So in terms of post-production, I mean, all of I didn't include it in this set of slides. It'll be part of the masterclass tomorrow, but I go through a little bit kind of the pipeline and how we navigate it to create science animations. The compositing techniques are really the same as for live footage. So there's a lot of, these images don't come right out of the 3D software. They usually come in multiple layers. We composite them together, sync them with sound, do color correction. We don't do all of the things that are usually done on live footage. So you mentioned things like, rotoscoping or things like that. We often can design that right out of the software. But then, I think a whole other area which is really of interest, and I already am hearing people and others in the field are doing this, is this software sits also upstream of game engines and other technologies. So rendering animations is one thing, but taking those models into interactive spaces, whether it's VR or game engines, is another exciting part of the work. And we saw last week examples of really the cutting edge of people doing this now in real time. 8K displays on domes, just amazing stuff. I mean, the future is here, kind of thing. So yeah, were there other? Yes, yep.
Question about the mental model, and I was having all this background, so I'm very beginning it's really that was missing, or maybe I didn't understand it as covered in another way. This piece would be the agreement that we all make about what
Say that one more time. So the agreement we all make not to include. Sorry.
what an octopus looks like.
Yes.
As a species or as a national.
Mm-hmm. Right. So I'm not sure if what you just said actually supports my argument or contradicts it. In other words, but I think right. I think so. The interesting thing, and if I understand what you're saying correctly, is this idea that there is, in the end, or we hope there's a shared agreement on what we mean by some of these concepts. And I followed up with some of the intentionally way more abstract terms in the life sciences, like from insulin to cell, to make the point that at the very least, when there are objects or things that we can rally around and observe together, it makes it a lot easier to agree, especially in individual instances, on what we're all talking about. What becomes, I think, a real problem is when, either as a small group of scientists or as an entire community, we talk about concepts, but we come to them with really very different experiences. Even though we're all biologists, let's say, we still come to them with very different experiences experimentally, in terms of how we study them, in terms of how we imagine them. And we're seeing...
Who knows how to activate the handheld mics? Yeah, I know, but then there's no...
Someone else is mic'd up. So we're actually really interested in this idea to see if it can help some of the inefficiencies we're seeing in the pharma industry, where you have these large, often geographically dispersed groups of researchers, of scientists. They're all aligned around the same goal, right? They all want to find, let's say, a small molecule inhibitor of protein X to cure a disease. But you have, molecular modelers working with computational biologists, working with clinicians, working with cell biologists, working with lawyers, and they all are trying to kind of grasp at the elephant in the room. And I think part of the problem, and I'm not saying it's the only solution, but part of the problem is that they come at the problem with very different mental models of what they're trying to solve. And what I'm suggesting in those examples is that visualization is a way to bring all the data into one environment, which it usually isn't. It's usually sitting in the different professional groups, and to bring all of that data and integrate it, to synthesize it, so that everyone can be looking at the same thing.
It's almost like you need a fake news checker, because at some point you have to get to the what we're talking about. So I think when you're talking about something that's interdisciplinary, you need, like when you're showing what your tools do, and you're saying you can have this tool that looks at it this way. I think part of the conversation has to be in visualization. So you're not just trying to get to one model, but you're trying to have a conversation.
Exactly.
Visualization.
Yeah. It's back to the all models are wrong. Some are useful. I mean, I think I know that sounds really simple, but it's actually, for me, that's always been a really powerful statement, because it's a starting point. It's never, and I didn't want to give the wrong impression that in doing some of those experiments about how to design the perfect animation, that's actually not the point. The point is, what are the design parameters that allows to achieve certain learning objectives? And that's the key, which is who's your audience and what are you trying to communicate? It's always context dependent. So I think another way of saying it is, if you're going to create these representations, you always have to explain where the data came from and what you did with it. I think it's the same problem that we're seeing for those of us who have children or are going through the educational system. There's this focus on science education about the facts as opposed to how we got the facts. And for me, it's the same thing in visualization. It's this notion of, here's a model, here's a visual model to the best of our abilities of the data. Now let's discuss what's missing. Let's discuss better ways to do it. But a conversation would be a great way to get at that. I don't know if that partly answered your question, or yeah.
Yeah, this is great. I mean, I can see there's going to be a lot to talk about later. I'm glad we're having a little reception after the whole talk here. And that'll be a great chance for all of you to continue the conversation. So right now I'm going to get back on with the program here, and let me just find the right thing here. There we go. All right. So let's thank Gaël again for a great presentation and his wonderful work. And I'm going to continue for just a few minutes to talk about what we do here at the New York Genome Center in terms of visualization. You've already seen a lot of proteins that looked a lot more impressive. This is the kind of stuff that makes it into scientific journals. And so I sometimes help out by creating figures for publications that sort of show the, actionable results. Here, these are all individual variants that we found through genome sequencing that make their way from the DNA into a protein that then has, all kinds of problematic issues, in this case for glioblastoma. Another way of looking at it is not just one protein at a time, but in displays like this that show you a whole range of genes. That's what those acronyms on the left mean, across a wide variety of samples. And we're trying to classify, all these combinations by whether or not they have certain attributes. So the blue rectangles are deletions, which is something that, again, happens a lot in cancer, that entire portions of DNA are wiped out, or amplifications where chromosomes get duplicated, or parts of them. And that causes all the malformations that you observe in cancer. Then another output from the cancer research is gene fusions. And these are some details here about a tool that we created to visualize gene fusions to help people understand better where the cancer wreaked havoc here, and where it created these new chimeric proteins sometimes that do horrendous things. And so here's a tool that sort of shows you how portions of a gene are chopped up. Those are the vertical lines that are connected by dots. Everything in between is omitted, and the outer portions are fused together. And this is some software that helps to tell geneticists and biologists what's going on in samples that came from individual sequencing runs. And then, as the last thing, I'm going to show you a short video from a tool that we built here to help biologists look at their sequencing data without having to have a bioinformatician sit by their side. So Metronome is a platform where you can upload your data sets, which you selected at the top. Then you start putting a query together that takes all the patient attributes that we fed into a database, things like, what portion of an experiment they came from. Then you can combine that with other criteria, like where the disease first was explained. You can chain these things together as much as you want. Then you can look at genes by position or select from lists. Or if you don't know anything about your data set, you can start your whole exploration by looking at a matrix similar to the one I showed you before, where you have genes listed on the left and then samples across the top. You can reorganize this, resort this however you want, and then make a selection across that area and start your query that way. So this is one way to look at a data set and start exploring it without any prior knowledge. The results kind of look like this. You get the list of genes that you just selected, and we tell you how many mutations were found in those samples. You see those in a table at the bottom. Then we also show them on a gene diagram, which unfortunately looks a bit crap on the projector here, but down at the bottom is the genomic sequence. So those are coordinates that tell you where the samples came from. And here we visualize all the patient attributes. So what age were they? You can select from a whole list of things that were all put into the database. You make those changes. and the diagrams all get updated. Then instead of just looking at it in sort of its distribution, you can look at the relationships that exist between the genetic attributes that you just selected and other attributes of the patients. So here we have on the right categorical dimensions, such as whether you were affected or unaffected. Are you a smoker? Yes or no? On the left are numeric dimensions. And there you can now start to look at, okay, if I just select the younger people, how does that other diagram change? How does my population change? And in the same way, when you scroll down, you would see that the gene diagram just got updated, and all those mutations that were there before might now be updated. So this is our way of basically getting at giving biologists a helpful tool to explore data that we're generating here, and thereby helping to move science forward, because it's going to hopefully democratize the whole process a lot more. And yeah, so with that, I'll finish up. And I want to introduce our next speaker, which is somebody that I've worked with in Australia for almost 4 years before I came to the New York Genome Center. Sean is somebody who is very passionate about visualization and has been involved in it, throughout his whole career pretty much, and has created a wonderful conference series that deals with visualization in biology called VIZBI. But he's the one that's going to tell you more about that. So from Sydney, Australia, from CSIRO and the Garvan Institute, here's Sean O'Donoghue.
Thanks, Christian. So I just thought I'd finish up and with a few sort of closing remarks and perhaps explain this mysterious term VIZBI that probably not everyone in the audience understands, but hopefully by the end of these slides you will. So as Gaël has shown us beautifully, as scientists we need to use visualization when we tell stories about our data, even if those stories occur on the tissue scale, which is relatively familiar, but things rapidly get very complex the closer that we zoom in. And certainly when we get down to the cellular scale, here we are down in the human microbiome, inside our digestive system, and showing some complex phenomena that happen there with soon after we eat. And then we get down to the molecular scale. This is a scale, as Gaël mentioned, that beyond the wavelength of light, it's something we don't really have good intuitions about, and so we absolutely need to draw upon many techniques and tools from visual storytelling in order to elucidate our discoveries. But as Christian showed, there's a whole other realm as scientists where we need visualization. That's not for explaining science, but it's for exploring scientific data. And this is an area that I spent most of my career in. And I'm just going to show in the next few slides. I'm not going to go into any detail. I'll just give you a quick glimpse into some of the latest visualization techniques being used in science. This is a review we're just about to publish. And we've cherry-picked some of the very most the latest visual techniques that are being applied across different areas. These are some different views of DNA data currently being worked on. These are views of RNA data where we're sequencing the RNA levels within single cells. That gives us tens of thousands, hundreds of thousands of different measurements from within each individual cell, and then looking at whole groups of cells and how they behave. So we have very complex behavior to somehow map down into a visual landscape. Protein structures and molecular structures in general, something we've been working on for a long time, but these fields are advancing incredibly, and the visual techniques are also advancing to keep pace. And then we have multiple molecules coordinating together into systems, and we need to get better at how we represent those as well. In some cases, these systems, as shown at the bottom, it's actually sort of a summary of events that occur in this case in a particular cell in response to a particular stimulus like insulin, and we have actually tens of thousands of distinct events that we can measure and track over time just within one cell, and again we can repeat those across multiple individual cells. And then we move up to the tissue scale, some really exciting things happening there. Perhaps that last one just at the bottom, the use of augmented reality with real-time surgery, I think is going to have an incredible, it's going to revolutionize surgery incredibly. It's already some fascinating stuff that's going on. And finally, if we look at the level of populations, things get even worse in a sense. Let's take the microbiome. So many of you probably know we have more cells in the body that are actually of bacterial origin than of eukaryotic origin. The genetic diversity in a human, if you look at all the genes inside of us, most of those distinct genes are actually bacterial, not human genes. And we're measuring those. And so there's tens of thousands of bacterial species within a human. Each of those has its own sequence, and somehow we need to represent all those data in ways that make sense, or at least what we can, as a scientist, make some sense from them and draw conclusions that will help human health. So we realized some years ago, to explore these data, there's enormous creativity required because the data that's being generated, in many cases, it's very new, never been generated before. And so then when we come to try to map those data, to visualize those data, explore those data, we've got to invent new visual metaphors, new landscapes, new ways of embedding these data. And we realize that although as scientists in the life sciences work across many different areas, we sort of have some sets of common challenges when we think about visualizing these data. So about nearly 10 years ago, we created a meeting in Heidelberg, Germany, as an experiment. We invited into the room the people who visualize data across these different fields, from DNA, RNA, proteins, cellular biology, tissue biology, and populations, the people that create the tools that scientists look at, put them into a room for 3 days. And actually, we didn't know if they would actually have anything meaningful to say to each other, but that first meeting was extremely exciting. There was a lot of cross-pollination of ideas across those techniques. And we also reached out to computer science, where you've probably seen many great TED Talks from computer scientists doing some very advanced visualization techniques. And we dragged some of them in, kicking and screaming into us biologists, because we have terrible problems to work on. And some of these computer scientists have fantastic solutions. The idea was to see if we could find matches between those two sets. And it was such a success. We managed to get funding both from Europe and also from the USA to run a series of meetings. It's been continuing ever since. We just had the 9th meeting last week in Boston. It was alluded to a couple of times. And here is the solution to the puzzle, the word VIZBI. It stands for a meeting on visualizing biological data. And, we came up with this idea that these tools that scientists are using to look at the data from DNA all the way through to tissues, through to populations, ultimately these tools should ideally talk to each other, because these different scales of biology are to some extent arbitrary. But the tools ain't going to talk to each other until the people that make the tools talk to each other. And there was no meeting before VIZBI where the people really who create those tools met. And so we're really proud. And actually, I think we saw particularly from this year's meeting that we've definitely developed a community of tool makers and also storytellers who are using each other's work in increasing ways. And we've had a big effect on the community. And actually, the 2016 meeting, we were fortunate to get written up in Nature, a short Nature article. And the editor came up with a really nice phrase, which I've sort of commandeered, which is that these are the visualizations transforming biology. And I think that's really true. How we look at data influences how we think about it. So to some extent, the people who create the tools that biologists use to view the data, they're actually transforming the process and the way in which we think about these data. So I'm not just telling you about this great conference that happens in other places to make you jealous. We actually have put a lot of work into making the outcomes of this conference quite tangible. So there's a website where you can get, for example, all the posters from previous years are there in Zoomable format. So in some cases, there's some really gorgeous examples. It's a great place to go to get ideas for an understanding of what's happening in the field. We've also videoed most talks, and people like Gaël and many others. If you've enjoyed Gaël's presentation, you should really give yourself a treat and go and explore the VIZBI video library collection. And then to explain the concept of the VIZBI showcase, we realized, well, VIZBI is really for us, the scientists who need to create visualizations to explore data, or who need to create animations to explain data. But what about for people who are just interested in science and want to see the latest science? And so the idea of the VIZBI Showcase was to create a series of evening events like this one, where we take people such as Gaël, who are focused on using visualization for explanation, and really sort of bring them to great places like this and allow them to showcase their work. So very happy to have done that. And Gaël, I think Christian also mentioned that tomorrow we're running a training course we call the Data Visualization Masterclass, where if you want to spend a day or 3/4 of a day, the three of us are going to be spending some time going through some of the tricks of the trade and explaining how we actually build these visualizations, both from the point of view of explanation as well as storytelling. And to give you also a bit of a idea and a glimpse into the future, if you're interested in this material, I happen to work in Sydney. I know that many people would love to go to Sydney but want an excuse. Let me give you some good excuses. So actually in the middle of every year in Sydney, in what we call winter, which is what everyone else in the world would call summer, the only thing for us in winter is it just gets dark early. It's still quite warm. We never get snowstorms like this morning. We have this incredible event. It's 3 weeks of the year. Every night they run these incredible projection technologies and project onto not just the Opera House actually, but the entire Circular Quay area. Essentially becomes a canvas. Now, I've been fortunate to collaborate with this group. It's called Vivid, and we've been running some events there. So essentially VIZBI Showcase-like events. So we're running a VIZBI Showcase with another molecular animator called Janet Iwasa. She's from Utah. We're doing another masterclass training event. So if you can't come tomorrow, you can come to Sydney. Quite convenient, right? And if you're curious about VIZBI, the next conference is next year back at EMBL Heidelberg in Germany. It's a really beautiful location. This is where we started. It's going to be our 10th meeting. We're thinking about doing some social celebrations. So please consider to come if you were excited by the material. If you can't come or just want to know more, sign up for the mailing list. We also have a blog. Please stay informed. So with that, I'm going to finish up and I'll hand back to Christian.
Thank you, Sean. Now we're coming to the final.
Should I switch back?
Doesn't really matter. The important thing is still on this USB stick, which are the lightning talks. You all might have seen on the invitation to this event that we were asking people not only to come and look at other people's work, but also to raise their hand and uncover all the activity that goes on within this community here. And unfortunately, I had a few cancellations, so it's not as plentiful as I was hoping, but I still need to move this stuff over. Hang on a second. Where are we? Sorry, just bear with me for 1 minute. Nope, that's not it. Oh. Okay, there we go. So if I can please have my 2 lightning talkers come over here. Basically, all you have to do is hit the left arrow to proceed, or right arrow actually in this case. And there we go. Speak into the mic, and Nika is first.
All right, good evening. My name is Nika Ford, and I'm an academic medical illustrator for the Icahn School of Medicine at Mount Sinai. So I'm just going to briefly go over one of my pieces. So this piece is the piece I did on the combo dual therapy stent used to treat coronary artery disease. And so what this stent therapy does is it has a luminal surface that has antibodies coating that luminal surface, and these antibodies attract endothelial progenitor cells, also known as EPC cells, which are circulating within your bloodstream. So once the stent device is coated with these EPC cells, it creates an endothelial layer, and the endothelial layer that it creates is the same endothelial layer that's on the interior surface of all the other blood cells within your body. So in that case, this luminal surface allows the stent to help protect the artery from thrombosis, which is blood clotting, and also prevents the abnormal narrowing of the blood vessel walls. So I just wanted to say that one of the things I find interesting about being able to combine art and science is the ability to really create some interesting visuals. For example, combining gross anatomy with molecular science in order to explain a complicated concept such as this one. So, thank you.
This image, this one belongs to me.
Mm-hmm.
Okay, I'm a postdoctoral research associate in NYGC, and I'm working with human-induced pluripotent stem cells. I convert them to motor neurons or astrocytes. So I work on, indeed, ALS, amyotrophic lateral sclerosis. I think all of you should know it. Stephen Hawking also had the ALS. So here you can see the astrocytes, which support the neurons, indeed. I acquired this image using confocal microscope upstairs. So the technique that I used is immunostaining. Basically, you choose your protein of interest, and then you find an antibody coupled to a fluorophore molecule to target it, and we excite it with certain lasers and we acquire this image. For example, here I image two different proteins which are expressed only in those astrocytes. When the protein that you see in magenta overlaps with the green one, you see it's like bright. I'm interested in the protrusions of those astrocytes. You can see it's here like a bridge contacting, two cells, they contact. Indeed, those are important to connect also with the neurons and with blood vessels. Those cells, they also regulate the blood flow. Yes, it's what I want to tell. I just want to describe you the image. In ALS, indeed, those astrocytes are important. The motor neurons, they die in ALS, but in Petri dish, the scientists, they showed that indeed, even if the motor neuron itself is healthy, if this astrocyte is sick, it can kill the motor neuron. So in our lab, we are working on the interaction of the astrocytes with motor neurons also. Thank you for your attention.
Thank you. All right. Thank you again, our lightning talk speakers. That was also one thing that we had started doing during these events in Sydney, is to just tease out people that sort of live in that intersection of art and science and encourage people to collaborate and get new things going. But since you had so many questions earlier, let's go next door into the cafe and continue the conversation over some drinks and food. Thanks for coming, you all.